Ekta Shukla, Associate Professor of the Amity University, Engineer Gautam Palani, CEO of the Genuine Biotech, Dr. Balakrishna Muttukuri, Chairman of the Mother Agri Biotech Laboratories, Dr. Chandrasekhar Biradar, who is the Country Director, Center for International Forestry Research and World Agroforestry, Mr. Abhay Gandhe, Advisor of the Transform Rural India Foundation, and my colleague, Dr. Shivkant Shukla, Deputy General Manager, BCIL. I extend a very grateful welcome to all our speakers and panelists. We are also heartened by excellent response from the tissue culture industry, academia, research scholars, nationally, internationally. We accord a wholehearted welcome to each one of the participants. Last but not the least, I extend a warm welcome to one and all. Thank you very much. Thank you. Over to you, Dr. Shukla.
Thank you, ma'am. Thank you for this setting the stage for this webinar. Now, I'd like to invite Dr. Rishi Kumar Tyagi, convener of the FCOP. Today, just informed that Dr. Ravi Khetrapal, Executive Secretary, he could not make it because some sudden preoccupation. So, I'd like to invite very senior expert, Dr. Rishi Kumar Tyagi, for the opening remark. As you know, Dr. Tyagi is the key person behind all the series of webinars. I've been telling this thing to all the audience. Dr. Tyagi is the coordinator of Asia Pacific Consortium on Agriculture, Biotechnology and Bioresources, APCOP, and he has done PhD from University of Delhi. He has served ICAR, Indian Council of Agriculture Research, in various capacities for 4 decades, 40 years. And he has made immense contribution in the area of agriculture and plant biotechnology. Credited 111 research papers, many books, 27 books, more than 70 invited lectures. And right now he is working for the sustainable agriculture development in Asia Pacific for the benefit of smallholder farmers by various initiatives. And he is also enabling many steering committee for policy development, advocacy, and a capacity building program. Dr. Tyagi, over to you.
Thank you, Dr. Shukla, for this generous introduction. And respected Mr. Chanjit Singh, Madam Purnima Sharma, Dr. Shukla, and distinguished speakers and panelists, and the participants who have joined virtually. It is quite heartening to note that more than 700 participants from 45 countries are registered. And I see more than 200 already there. And this webinar is being live streamed on YouTube also. I don't know how many are there. So it is quite a successful venture to organize this series of webinar. So before I start making some comments, opening remarks, I also joined Dr. Purnima to welcome Mr. Chanjit Singh, the Additional Secretary of Ministry of Rural Development, Government of India. I welcome Madam Yu also on behalf of APARI and on behalf of Dr. Ravi Khetrapal and on my own behalf to this webinar. And I think BCIL has done an excellent job as a partner to APARI to conduct these webinar series. And already, as you have mentioned, 2 have been concluded. This is the 3rd one in the series, and 4th one on the ornamental crops will be held on next month on 26th August. Dr. Shukla has done an excellent job by contacting the expert speakers as well as the panelists to make it more vibrant. And since this current webinar is dedicated to the tissue culture of tree woody plants, particularly on bamboo and teak, with the significance, best practices, and way forward. Mr. Chairman, this in fact webinar series was organized for the benefit of the countries of Africa and Asia. And before that, let me take a minute to tell about the APARI, what we are doing and what we are. It is mentioned that APARI Asia Pacific. I got some problem here. Okay. So APARI is the Asia Pacific Association of Agriculture Research Institutions, which is located in Bangkok, Thailand. In fact, it was established by the Food and Agriculture Organization of United Nations in 1990. So you can say it is almost 32, 33 years old organization. And it has its own vision and a very well worked out vision 2030. And under that vision, we are working for the sustainable development in the Asia Pacific region. APARI has a member-based country. It is a political non-profit organization. And currently it has about 82 members, which are located in 32 countries in Asia Pacific region. Some of the CG centers are also the members of APARI. So we are working for the members and we are working with the members. So in this way, BCIL is also one of the reciprocal member of APARI. That's why we have developed this partnership, very fruitful partnership, in the same spirit that we are working for the members and we are working with the members. Here in this webinar, I am very thankful to the C4E CRAF also, and Dr. One of the very eminent panelists, Dr. Chandrasekhar Biradar, is also with us. He belongs to C4E CRAF, and C4E CRAF is the one international organization where this agroforestry and the tree or woody plants used for the sustainable development or the enhancing the income of the rural farmers or smallholder farmers at the global level. So you listen to him, you will have the opportunity to listen to him. Two more points I want to mention that we have, as I mentioned, we have the 4 thematic area. One is the natural resource management, risk management, including development and value chain integration, and policy development and advocacy in the Asia Pacific region. So as I mentioned, under the 2030 vision, we are having these thematic area, and one under the natural resource management, the EPCOAP, Asia Pacific Consortium on Agriculture Biotechnology, which was established again by FAO and APARI in 2003 to promote the biotechnology and for effective conservation and utilization of bioresources in Asia Pacific region. So this is all brief about the organization. Dr. Purnima has mentioned in detail about the webinar series and this webinar. I need not to underline again the importance of the agroforestry or the tree or the woody plants, but certainly a few words I would like to mention about the tissue culture technology, because this is also my very favorite subject, and I have put in my almost the 20 years of my career in the tissue culture of the different crops. As a biotechnologist, I have been attending a lot of argument and a lot of counter arguments that the scope and the benefit of the biotechnology. Maybe other biotechnology tools may not be the convincing to everyone because there are a lot of controversies, and in fact, forced controversies are created sometimes by some interested groups or lobbies. But tissue culture is one of the part of the biotechnology which has been proven undoubtedly, unanimously, and unambiguously the whole benefit to the whole humankind, to providing the quality planting material, which you will be listening in this webinar about the particularly woody plants, is the only technology, and it is very, very well established and very well accepted technology without any controversy at the global level. So, Mr. Singh, we organized this webinar series on the request or the feedback of our member countries, which are 22, including India, in the Asia-Pacific region. And other than member countries, we have some other members, other category members, about 80, 82, as I mentioned. So on the basis of their request, this webinar series is organized. And we selected the groups of the plants which are of the interest of this region to the smallholder farmers to enhance the trade, to enhance the livelihood or improve the livelihood of the smallholder farmers or improve the trade between the countries. I think this seminar will cover all these aspects on the woody plants in general and bamboo and teak in particular by the eminent speakers, which we had today, Dr. Shukla and Dr. Balakrishnan, and the three eminent panelists are there. So I'll not take more time between you and the real speakers, because I'm not the expert in agroforestry. I may be the expert on tissue culture, but I am not working in agroforestry. So let us listen the speakers and the panelists and the discussion or this webinar. With these words, I again welcome you all and thank Mr. Charanjit Singh for accepting our invitation, these speakers for accepting the invitation, and BCIL, particularly Dr. Purnima Sharma and Dr. Shukla, who have been very kind enough to shoulder this responsibility in partnership with APARI and making it a great success. So thank you once again, and over to Dr. Shukla.
Thank you, Dr. Tyagi, for your kind words. You're also providing the detailed background of this program. Now, we are eagerly waiting to listen Mr. Charanjit Singh, Honorable Chairman of this webinar. Mr. Singh is a IFS of 1990 batch and is a Master of Philosophy in Strategic Studies from Madras University. He has extensive experience in the area of extension and working toward the environment. He earlier served as a Director of Department of Land and Resources, Government of India from 2008 to 2015. He is heading a very important mission of Government of India, that is a National Rural Livelihood Mission. And when we talk about bamboo and teak, it becomes highly relevant. So, Mr. Singh, thank you very much for joining this webinar. And even after this webinar, we would seek your kind support to carry the recommendation forward. Mr. Singh, over to you, sir.
Okay, thank you very much, Dr. Shivakant Shukla ji, for giving me this opportunity to address such a, what should I say, illuminated speakers and other guests on this webinar. And I wish a very good day to everybody in the beginning. So it's a really nice thing that you are addressing this very critical component of this tissue culture. So I wouldn't go into the science of the tissue culture because there are so many experts here. So, but I know that what is the significance of the tissue culture? As you must have read about these biofortified crops nowadays, then these are pest resistance crops. This has been all due to the advances in the tissue culture, and a lot of work has been done. So rather than addressing the issue of tissue culture, I would address how it is relevant to the rural India and to the rural poor women with whom the Ministry of Rural Development is working through this very ambitious scheme of National Rural Livelihood Mission. So I will just give you some figures that we mobilize one poor woman from each household from across the country. So far, we have mobilized 83 million households. So you can see this is a humongous number. And we have formed 7.7 million self-help groups of these women. So but we believe that if the women work in group and they try to address the issue of poverty on their own, then this is very effective and sustainable also, and then succeed also in this direction. So that is the big picture on which we are working. And here, as you mentioned also, teak, bamboo, etc. play a very vital role. Not only the people who are living in the forest area or in the forest fringe area, but beyond the forest also. Because these are very good crops and a lot of money can be earned out of that. But due to not having good plantations of these things and degradation of the existing crops due to various regions, the returns from these crops are very, very low. So here the tissue culture comes into picture, that how we can address these issues. And as you will be happy to know that we are striving that all of these rural poor women, that is 83 million households, should come out of the poverty, and then they should have sustainable livelihoods. And we are trying to provide them different sorts of livelihoods. And one of our models in that is of the bamboo plantations, because bamboo is used in many ways, not only as a food, then construction material, furniture, handicraft, so many usages of bamboo are there. And it plays a very critical role, especially in the northeastern India. So we feel that bamboo has got a very important role. And we have kept a target that at least 1 million livelihoods will be generated through bamboo only. So that is our target. And for this target, I think tissue culture is going to play a very critical role. And you will be happy to know that we have not only mobilized these households, we have placed community resource persons in the villages, those who provide 24/7 hand-holding to our rural poor women. So their number is 360,000. So this is also a huge number. So they are trained in various thematics, whether in agriculture or animal husbandry or anti-FP. So many activities are being taken up by them. So I feel that these community resource persons can also play a very important role as we move ahead in this direction. So what do I feel are the constraints that why we are not making the progress as much as we should make? So I jotted down some of the points after giving a thought yesterday. The first point I feel is the awareness. There is not much awareness in the rural hinterland of the country that what is the impact of the tissue culture and how our forestry, trees or plantations can be improved. For that, I feel that we should have a good awareness generation program. And this awareness generation program, I feel that should not be only in English and Hindi only. We need to go to the regional languages also, because those are very important. Few days back, I went to one interior districts of Madhya Pradesh, that is Dindori in Madhya Pradesh. So it's a very remote area. So if we want, and I was surprised to know that the people were listening to the community radio in the Baiga language. That is the tribal language. So if we want to reach such people, I think we need to speak in their language and we need to address these issues. And for that, I feel that we need to prepare the IEC material also, whether these are videos or some other sort of literature is there, where the benefits of this tissue culture should clearly come out so that the people know about that. And as I come from the state of Punjab, I know that how the agricultural revolution took place there. They demonstrated the different crops, that how the return from these crops can be taken. What will be the yield, what should be the frequency of watering, etc. So that whole revolution took place because a massive campaign took place in this direction. So that is my point. And the third thing I feel is the cost effectiveness. Cost effectiveness also plays a very critical role. And this also needs to be demonstrated to our stakeholders so that they can take up these things. And the next thing I feel is the hand holding. Because if we are teaching something to the farmers, then 24/7 hand holding as is being provided by our community resource persons. That is also very important. because we have seen that one bad experience gives a very bad publicity to the whole thing. So the continuous hand holding is important so that the people know about the results also, and they can take up these things on a large scale. So these are the some of the points which I jotted down yesterday, which I feel are very important, and I feel that these will be deliberated along with the science of tissue culture. I have no doubt that these also needs to be deliberated upon. So I wish all the best to the webinar today. And as you know that the parliament session is going on, I was busy in the morning also. So I would have to leave within 5-10 minutes again. So thank you very much for giving me this opportunity.
Thank you so much, Dr. Singh, for this valuable insight and gracious presence. In spite of all the busy schedule, because this session is going on, and we know how much busy you are, all the senior bureaucrats are so much busy. So thank you very much for highlighting this issue of rural livelihood and community development. Important aspect you highlighted, that is creating awareness in local language. And the BCI did for this culture program in 5 years back. For all the state government, we did regional level program, we translated all the documents. So we have done extensive work to promote this culture work in local and regional level. So again, I think we'll discuss this with you, and we'll seek your guidance how to take it forward. So thank you so much, sir.
Thank you.
So now we'll move toward the technical session. How we have planned, the way it was structured previous event, we'll have one academic presentation from the key researcher highlighting the key aspect of this culture. Then we'll move toward the industry presentation, giving the success story. So now we'll go for academic presentation by Dr. Susmita Shukla, who is associate professor in Amity University, and she has long experience in working in the area of plant culture for last 20 years. She has done extensive work in embryo rescue, secondary metabolite production, and particularly clonal propagation of tree species. And she is recipient of many awards like TBT, Biocare, INSA Nasif Fellowship, and many more. So, over to you, Dr. Susmita.
Thank you so much, Dr. Shukla, for giving such an introduction. And very good morning to everyone. So let me share, if I'm audible, am I audible enough?
Yes, please share your slides.
So let me share my slide first, and then directly I will be moving towards my presentation. I hope it is visible.
Yes, visible. Click, make it slide some more.
Yeah, just a second.
Yeah, fine.
A very good morning to one and all here. And first of all, I would like to thank you, BCIL and APARI, for giving me opportunity to speak in this international webinar, specifically with respect to tree tissue culture. And myself, Dr. Susmita Shukla, as I've already said, I'm working in this plant tissue culture technology since long, and we have also regenerated different protocols related to the tree and different woody plant species. So I will be highlighting the research aspect, what are the different things which being researcher we face, and the way forward how we can cope up with this. As we know that world is a home about more than 60,000 of different trees, and the importance has been already taught by Dr. Singh and Dr. Tyagi, who has already said that yes, tree tissue culture and the tree importance of tree is very much beneficial in all respect, not only with respect to clean air, water, biodiversity, but also it helps to regulate the climate. And we know how the climate is being just at in the global warming, we can say how it is affecting the climate. So we are losing an area of the forest, and it has been mentioned. And the thing is that how we can stop these deforestation. These tree and plants are being degraded and logged at astonishing rate. From this study done by Crother TW 2015, we came to know about the global number of trees, which has been fallen by approximately 46% since the start of human civilizations. So there are many different things, many different cases which happened, which are still in continuation, and we are losing the trees at a tremendous rate. So we need to understand the solution regarding this. And the solution which we are doing with, which we are working, is the micropropagation trees on or the woody plant species, which is very hard to regenerate. But yes, being tissue culturist, many of the scientists has worked in this field, and this has been given very good implications in the mass propagation and production of the tree species. Through this, as it has been said, that tissue culture has been already proven to meet all the challenges. Specifically, when we talk about the mass scale propagation and of different tree species and the woody plant species, we can give rise to the different elite clone planting materials. But before we start for going for micropropagation, as we know that micropropagation, the technology is, we are propagating large number of plant species from a small explant. Yes, it is the most, you can say that the boon which is being, is there in the plant, the totem, because of its totipotency in nature, it is having that particular capacity to regenerate into the whole plant. But before, when we go with the tree or the plant species, different challenges we need to face, different problems we face, which we need to overcome. And of course, once it has been done, we can get different opportunities which will be helpful for the social benefits. As I said, that I will be talking about the problem which are being faced in vitro propagation. I'm looking into the academic work and the research work which we have done, and which the other scientists also face. The major problem which are being faced by this diagrammatic explanation of the slide, it's very much clear that the contamination is the important or the strongest problem which we face when we move towards the woody plant tissue culture or the tree propagation of the tree species. But unless you know that in vitro condition, it is very difficult, because the endogenous microbes are there, which we come to know about it after a long time. It's not the same thing as it happens with the other plant tissues, or the when while one is working with the herbs or different kind of shrubs on it. But in this case, it has been seen that endogenous microbes comes on after a longer period. When you go for subculturing one or the other area, then you can understand that yes, this is thing which we have not thought, but all of a sudden it came. But there are certain ways which we can, if we know, and we can combat with those problem as well. So in solution, I have already mentioned the chemicals which can be used to eliminate those contamination. Even certain antibiotics and fungicides are also there. And in the last report, I've already mentioned one particular media has been made which has encountered and found very effective for combating with the endogenous contaminants. The second problem which is there is the recalcitrant. Recalcitrants you can define as inability of plant cells and tissues to respond in an in vitro environment. And tissue recalcitrants generated because of the reaction of free radicals with the macromolecule of cells, causing cellular dysfunction. But yes, this can also be we can fight with these problem also if we are having a proper planning. And to avoid this, the important thing is that because tree are the mature plant, tree species are the mature one, it's very rigid enough to grow into the in vitro condition. We need to think about which kind of explant will be the best to give result, to get result. And here you can see that how we can avoid it. And the thing is that by explant selection, you need to select the juvenile part which can be there. And certain reports has also been stated that by spraying the cytokinin in it, it can also overcome those recalcitrants in nature. Now, the third important thing is the browning, that is due to the extraction of the phenolic compound which is already into the plantlet, and in the media it comes out, and the media get dark brown in color, and which is toxic to the tissues, and it hinders the growth of the plant tissues which you want to propagate. But there are certain ways, as I've already said, that this browning can also be combated by different ways. For example, by using different adjuvants, activated charcoal is there, PVP is there, ascorbic acid is there. But yes, you need to decide you need to identify which adjuvant or which chemical is working, or which synthetic adjuvant you are using is working with respect to your species which you want to grow in vitro. One of the example I have seen that that is a combination treatment, combination of ascorbic acid and citric acid has been taken, and it has been found that yes, it is being found suitable for browning issues. And then there are also activated charcoal, as I've said, that certain species are already being using, and it is a very good adsorbent where the phenolic exudates are being adsorbed by those compounds. The next is the vitrification. Vitrification is again another important problem which are being faced over here, and we need to understand what this vitrification is. So it's a physiological disturbance or malformation occurring in tissue culture plants, and it directly affects the proper growth and development. What happens here? The plants just turns glassy, and thick and enlarged and brittle leaves and stems are being formed, which we don't want being a plant tissue culturist. So again, there has been beliefs that yes, these are the factors which are there, and certain oxidative stress factors are being maintained. But the solution, again, we need to understand, yes, there are problems for so many things, but we need to understand the solution, and we need to find out how we can overcome those problems. So here, the solution I have already written. You need to judge whether which kind of carbohydrate level is working. Sometimes it may happen the level of carbohydrate which you are taking is not fitting into it, and for that you need to understand this. And by changing the light intensity, this has also been happened that yes, the tree species is not getting the proper light intensity. For that also you have to work on. You can even modify the concentration of agar, you can check it, and humidity as well, and certain papers has been there where they have mentioned about the calcium concentration as well. The next which we face is the acclimatization. Acclimatization plays a very, very important role. And specifically when we talk about the threatened and endangered plant species, we have to be very careful because it is already in that particular category. We need to protect and we need to acclimatize and make it harden so that it can grow properly. So again, here also I have quoted and listed some of the points through which we can combat with this particular acclimatization, that what we have to be very careful while we are going for acclimatization or hardening our plantlets, because what you're doing, you are just growing all the plantlets in vitro condition, in the lab condition. Now you will be forcing that plants to grow by itself into the field. So there again, it has been seen that when you're going for the tree species specifically, it has been seen that if we go for the elongation medium for a little more longer period, then we found that it helps in the acclimatization purpose. And second thing, peat pellets are being used soaked in water for rooting substrate, cutting off the callus and planting plantlets in low intensity after the rooting. This has been seen that it has been useful for acclimatization. Now, this was the problems which was there. I will be coming towards the opportunities as well. As Dr. Singh already mentioned that yes, it is affecting it. They are looking for the livelihood of the all rural and urban areas. It is an important part that we should understand, and we should create awareness about the tree propagation overall in those region as well. So I will be taking few points where we can understand the importance of these things, which can, if we are able to go for this, yes, we can help those people to earn their own, to generate their own income. So as I have already mentioned here, that agroforestry has significant potential to provide employment by not only awareness, by creating awareness, and if they start using those things, yes, they can earn and they can generate income by implementing this tissue culture technology as well. Agroforestry, I've already said that employment opportunities are there, and capacity building for farmers. This is again another important thing. If we farmers are being trained and they are giving all the different information and the technical help to them for this type of growing these for raising these tissue culture tree species, it will be a great help for those people as well. But and another important point which I want to focus here about the indigenous tree propagation. We are, being in India, we are so rich in different biodiversity, and the indigenous tree species are so many things, and the need is there. We need to propagate those at much higher rate. So therefore, I have mentioned that yes, indigenous tree propagation, if once can be done, it will be of great help. Certain more points are there. I will not go with this. I will be directly moving to my next slide, speaking on the teak, the Tectona grandis. As it has been already said that it is a timber plant and we are knowing it. But other than timber wood quality, it is very rich in medicinal properties also. It is having the huge implications, which I will be talking to you later, but let's go with the tissue culture of the teak. How and why tissue culture, and what are the important protocols which has been developed by the different scientists overall worldwide. Some of that I will be quoting here to make to understand that yes, these are the things which has been already done by different scientists, and if we want to go for propagation of such thing, we can move ahead. Otherwise, our industry speakers are already present there who will be sharing their own experience related to the commercialization. As we know that R&D and commercialization are two complementary to each other, but yes, we need to make a balance between both of the space to move forward. Now, teak, as I've already said, it is a most prized high value. Everybody is knowing that. So, but I will be talking about why teak propagation, tissue culture teak is required. As traditionally reproduced through seeds, we know that germination is a little bit difficult because of its hard seed coat, low seed quality, and late seed production. This is what. And micropropagation is the next approach to overcome this issue. That is a very thing which we can overcome with this, whether it is a seed coat or low seed quality. Yes, by tissue culture technology, we can combat and we can produce this disease-free planting material in a large scale. As I've already said, that is the need for need of an R for the tissue culture. By tissue culture, the conventional methods which was applied, still people are using it, but I can say that they are slow and time consuming as compared to the plant tissue culture propagated plants which can be done. So these are the genetically improved variety if we are selecting the strong mother plant, the elite or the plus cultivars. And this will not only help in conservation of elite germplasm, but also we can restore restoration of afforestation can be done, and ultimately it will be creating a monetary resource. These are some of the protocols which has been developed by different scientists, which I have mentioned over here. As Tiwari et al. In 2003, different explants has been taken. As I've already said, that explant selection plays a very important role. And here you can see the different scientists have been working, different groups has been worked on this species, and they got the different result as well. That depends on what kind of plant species they are growing and which one is working for you on. That is plays a very important role. For example, growth hormone, you can see Tiwari et al. They have used the BAP and IA combination, whereas in Kozar and Shazza, they will say they have taken the Kinetin and TZ. So 78% was there, 77.9% they have got the survival rate. At the same time, when we go for Mendoza de Caires et al. In 2007, you can see the 100% result of multiple shoots has been given. That is the important thing that people have done in so many different places, different areas. But the thing is that at what scale it has been reached, that we need to take care of. Sorry. So this is what we have to understand, that what we are going and how we are moving towards this tissue culture technique. You can see here the different Rita bioreactors has been also used. They have proven that 30 initial explants double the 8 times, but that depends on the probability of the species which is there. These are the different glimpses of the in vitro method of plantlet regeneration. The overall pictorial or the description has been given, how we are sterilizing, how the mother plant is being selected, all the different stages, the initiation stage, the multiplication stage, then moving towards the rooting, and finally to the acclimatization. Everything is being mentioned, and this is the overall cycle which one need to complete if you want to go for the in vitro protocol. This is the large scale production which is already being given, and yes, it is being successful, which will be carried forward by our speakers from different industries. Now, as I've talked about the health benefit of these three species, teak is renowned for its durable wood. We already know it is being used for different purposes, furniture, flooring, chips, and etc. But you need to understand as the quinones which is present in the sawdust has shown the resistance against the fungi, and it's an ideal for many purposes in tropical condition. It is not only that the quinones is present, it is also known for its antioxidant and anti-inflammatory properties. And it has been also seen that it is cure the cold, promote growth of new hair, so and so. So there are medicinal benefits also. So tissue culture is not only the propagation of the plant, but we can go for the secondary metabolite production as well. We can enhance those particular compound in the plant, and then again it can be used in the pharmaceutical industry as well through tissue culture. These are the teak products. I will not go with the teak products and all. It's already known, but I want to just explain about the medicinal and the economical value. If we want to go for these kind of thing, yes, we have to work thoroughly. We need to understand, we need to do lots of research before we start working on these three species. Moving towards the bamboo, as we know that it is a non-timber forest product, and it is having a quite glorious history behind it. And because of its demand, it is being known as green gold as well. There are so many varieties of this, but again, I say that if we want to go for tissue culture, then why tissue culture? We need to understand that what is the need of tissue culture, why we want to move towards this particular direction. As in teak also, I have said that what was the major challenges which need to be faced. The similar happens here also, but here you can see that sexual reproduction is not reliable in bamboo species, and due to the long flowering cycle and low viability rate of the seed propagation, it is difficult. So therefore, we say that yes, tissue culture is required. Here again, the importance of tissue culture has been given, not the tissue culture, but starting just the bamboo. If we see that it is the non-timber wood products, but the other things are also equally important thing here. Many other places in Asian countries, we know that it is being used in preparing different delicious dishes, including soups and curries. Even the different artisans are also going for different preparation of crafts, and different jewelry, home decor items are being also made. So I want to just say that yes, industrial application is also very important for these particular species. Again, I went through the different papers where I came to know about the in vitro protocol for different bamboo species. Here, Sineton 26 from Nigeria, and then here they have done the different tissue culture. Ansari et al. 2017 also they did with, and Guantan in 2018 they have also gone with the tissue culture of different bamboo species. Yes, I want to say that yes, the protocols are there. In the reports, people are working on that, but when we want to go for the field, it may work in a separate way. That is how you need to go for this commercialization purpose. Until commercialization is being done, it is very, very difficult to understand how and what for your work is. So I will be going a little bit faster. Here is the different regeneration protocol which was there. Let me check upon if I'm let me know if if I'm not taking much time. I will try to complete at first as is being said. Here you can see this is the explant preparation sterilization. This is the overall technology which is being done, which is through which the in vitro grown plantlets is already there. Here the industrial applications, the woody plant species are already there, bamboo charcoal and the other products are being mentioned, which I've already stated, so I will not go. These are some of the bamboo products which are available here. Now coming to the tree species which are being worked, and these are some of the woody plant species which we have worked on and taken from the different agroclimatic zone which has been covered from various zones, and this work has been done on Folia erecta, Olea europea, then citrus species, Musa variety in Vigna, but I will be speaking on the Citrus macroptera. Here you can see that Citrus macroptera is fallen into the endangered tree species, which is very much required. We need to it is having various applications. And different delicacies are also being thing. Again, the tissue culture of this woody tree species has been taken. The lacuna in conventional method was 6 to 8 years of generation gap of this woody perennial crop and difficult to grow. But here you can see the micropropagation of these endangered citrus species has been done. These are the unpublished data of ours, where again has been done, and the tree has been successfully propagated, and it is in the final stage now. Another important species which we have worked on is the Punica granatum, and that too full erecta. And these are the currently TC rays Punica granatum fruiting and flowering at the present. And this is having its own importance. These are the different stages of the Punica granatum which has been done. And yes, these are the actual preacher which recently taken and has been fully hardened, and plant is already there. Again, another important thing which I would like to highlight, that is Olea europea, that is the olive which has been worked. The cultivar is Barnia. No one has worked on this, and in the picture itself, it's clear that how big the tree has been grown, and now the it is having a just we are waiting for the fruiting of this. These are the in vitro regeneration of Olea europea. Unpublished pictures are there. Still the work is going on, and we are working in the various other field as well. And the micropropagation of forestry species, as in the earlier stages, I've already talked about the rapid deforestation and depletion of genetic stock. You can see, you can check that how it has been done. So I will be running through two different species which we have worked on, Stereospermum personatum husk, which is again a tree which belongs to family Bignoniaceae, and it is being used for preparation of Chyawanprash and Ayurvedic tonic. Everybody is knowing about the Chyawanprash, so I will be not moving with that. But other than that, it is also having a timber wood quality. It has been used for house building, tea boxes, matchboxes, crafts, flowers, etc. These are the different steps. These are the different steps where it has been taken forward about the Stereospermum personatum. Second is the Stereospermum savalens DC. This is being used for the Ayurvedic formulation known as Dasmula. And in Dasmula also, we know that it is an Ayurvedic formulation. It is used for clearing the migraine, antidepressant, astringent, and liver stimulant as well. These are some of the real pictures of the work which has been done. And with this, I come to a conclusion that biotechnology has the potential to be part of the solution in protecting forestry species and the other woody plant species. And considerable investment is needed to further basic understanding of tree genetics and the effectiveness of biotechnology in mitigating forest threats and impact on ecosystem. And the micropropagation, firstly, it can be a little bit difficult, but once it is being standardized, it is the most important thing which can be taken up for producing genetically identical individual, the quality plant and material. And it is the right and smart R&D technique for rescuing extremely valued tree species, specifically in developed countries for Asia Pacific region, which we are talking about. At the end, I would like to thank you, Dr. Ravi Khetrapal, Dr. Rishi Tyagi from Apari, our MD BCIL, Dr. Purnima Sharma, Dr. Shivkan Shukla, DGM BCIL, our Honorable Founder President Sir, Chancellor Sir, and Vice Chancellor Ma'am from Amity University, and all the eminent speakers, panelist members, and participants for the patience listening. These are my team and co-research team. Thank you. Thank you all. Hope I have not taken much time.
Thank you, Dr. Susmita, for this detailed presentation. In fact, before this webinar, many registered participants, they asked many questions regarding their constraint while they're going for the field structure, like high endophytic contamination, survival rate, browning, phenolic excretion. So I think you took all the points very nicely, and whenever there is query, it will direct to you. Also, you also covered that other than teak and bamboo, there are many tree species, perennial species, which are very important, like citrus, olive, or like that. So thanks for this comprehensive presentation. I hope audience will enjoy this thing. So before now, we'll move toward the industrial presentation. This is very important. We talked about research aspect. Now we talk about the commercial aspect. How industry has witnessed this success. Because unless until it is reaching to the ground, to the field level, the science will not witness the great success. So before we move for this industrial presentation, we have a brief poll with the audience so that we'll get their feedback. Just like other webinar, we have 3 questions. They will take just 15 to 20 seconds to answer that. I request all the participants to respond to this query. I request my colleague to flash that poll question. There are three questions. Just you need to select only one. You might have received this poll question on your screen. The first question: do you agree that tissue culture technology is better than conventional method for the production of tree or woody plants? Strongly agree, agree, or disagree. You need to select one. Second one: have the plant tissue culture techniques been optimally deployed for the production of tree woody plants in your region or country? Yes, no, or not sure. Three options. Now the third question: do you agree that there is need for an effective training program for promoting entrepreneurship in tissue culture of tree or woody plant in Asia Pacific region? So just three specific questions. Select one option out of given option and submit your query. So just wait for a few seconds. Then we'll end the poll. Last 5 seconds. 5, 4, 3, 2, 1. Now we'll close it and we'll just display the result because we have a large number of participants for 45 countries, we told, large number attending. So this is the result. That majority of the audience, they strongly agree, right, or they agree that tissue culture is the best option to propagate. Now the second one, they feel that this technique has not been optimally used. Many audience they say that, and many of the reasons I think this has been used successfully. So it gives the success of this technology, and 98% say that there is a need for an effective training program in the Asia Pacific region. So that is a recommendation. I think we need to take it up further to provide hands-on training and creating awareness, as Mr. Singh told. Please end this poll result. Thank you so much for participating, all the audience, on this poll. Now, as I told, we'll move for the industry presentation. And first, we'll have presentation from engineer Gautam Palani. He will present about the success of bamboo tissue culture. Basically, Mr. Palani is a mechanical engineer and he is working for many years for the tree tissue culture, and not only tree, banana, ginger, turmeric, many crop is propagating as chief operating officer of Genuine Biotech. He's a young entrepreneur, as you can see, and he's leading his team, young team, and providing quality material across the country and also exporting plants in different regions. So being a tissue culture entrepreneur and bamboo is very important for this program. Mr. Palani, over to you. Please share your slide.
Yes, sir. Thank you. Thank you, everyone. Thank you, Apari, and thank you, BCIL, for inviting me as a speaker. It is very honor to be in this part. There's already 275 participants, and very lucky to talk in this. And as we talk about the commercialization of bamboo, the success story, it is not an easy one. It took us around 3 to 4 years for to standardize. And it was a great help from Dr. Shivakant Shukla. So he has helped us a lot in collecting good material, as well as he supported us a lot from BCIL. Thank you so much for this. And now I'm going through the technical part. So the slide is visible?
Yes.
Yes. So we Genuine Biotech, we started on 2006 and 7, and it's almost we are into banana, then bamboos, ginger, turmeric, and many ornamental varieties. But since 3 years, we are concentrating mainly on bamboo, since the market is very high and it is the present need in the world for climate change. So we are into that. And these are our recognitions. We are recognized with DBT and many institutes as well. And yesterday we are awarded for Z News National Achievers Award for we are working for climate change. Luckily, so yesterday night we were at Delhi, and today we are here.
Congrats.
Yeah, and the thing is that we know about tissue culture. So what is the use? And there is no doubt that the tissue culture is success everywhere, and it has been in the market since 20 years. But why for the tissue culture is needed for bamboo? So that is very important topic now. So we tried with many seeds collection and all, but finally the mortality was around 95% in the seeds. And also the seed availability is very, very less because the bamboo flowering is not prominent everywhere. So we have to wait and see where the bamboo flowers. So we should wait for that. In case if you are looking for these rhizome plants, so we don't know the age of the plant. So what is the passport data? So we don't know when it may flower. So we cannot easily choose a rhizome plant as well. And also one more thing in rhizome plants is that it may contain virus, bacteria, and many fungal diseases. So we cannot use that very easily. And also this bamboo propagation to this vegetative method is very, very costly. We cannot produce in lakhs and lakhs to supply to the government or to the big farmers. And also the vegetative method is too costly, and also it is not cost effective as and also it requires too much of area. And in terms of seeds, if you see, we cannot easily identify the species of the bamboo by seeing the seeds. In this way, so the many people are giving wrong seeds in name of different species. So these are the main disadvantages of the seed as well as the vegetative propagated these bamboo plants. And what are the advantages of tissue culture bamboo plant is it is true to the type, and it has high market value, high yield, and high quality. And also throughout the year, the plantation is possible because the tissue culture process is happening all over the year. So we can supply at any time, at any month. And also, since the mother plant we are testing, we are taking is a disease-free and virus-free plant. So 100% of the plants what we are going to give should be the disease-free and virus-free plants. And it is genetically pure. And the uniform growth will be there. This uniform growth we can easily identify in banana. So in bamboo, we cannot easily identify, but in banana, we can easily identify. And mass production at less time, at less cost, then it is season independent. So now, this is the main point in the selection of this bamboo mother plant. So unlike this banana and other variety, what we seen in the bamboo mother plant is that we used to collect different mother plants and give testing. Even though the virus and the bacteria is negative, though their performance inside the lab is very, very bad. If, for example, if we take around 50 healthy mother plants, in that only 2 or 3 mother plants will give us more multiplication ratio, and it will come. It will perform better. So this is one of the main thing. We should not choose only 1 mother plant or 2 mother plant. We have to constantly have 50 mother plants or 100 mother plants. From that, we should identify which clone or which mother plant is performing well. From that mother plant, if we do, our multiplication ratio and the performance will be too high, even though the virus and disease are negative. And mostly in our company, so we are having a seed germinated plant mostly as a mother plant. So whatever the plant which has the seeds, for Bulgova there is no seeds, for Stocksey there is no seeds, but other plants of bamboo, we are searching for a seed germinated plant. From the seed germinated plant, we are growing it for 1 year. After 1 year, we take explants from them. Such a way, if we take the explants from 1 year old plant, then the tissue culture plants what we are producing is also it's equivalent to 1 year old. If we take explants from a 10-year-old plant, if we do tissue culture, then the outcome of that will be also the 10-year-old plant. It is more soon it may flower. So after we take the mother plants, we will ensure the morphological and physical characteristics matches to the particular species. We'll see the clump height and clump diameter, clump thickness, so number of clumps. So everything will identify. So then we will we'll see whether okay this is the real Bulgova or real Tulda, and we will choose the better Tulda. For example, in Tulda, some plants will be of 50 to 55 centimeter of introrder distance. Some plant will have up to 60 centimeter introrder distance. So we'll choose that better one always. So this is the main thing. We initially went to many places to see the bamboo plants and collect the proper explants, but finally it failed everywhere. So what we did is that we allotted a space in our company. We are having around 49 species of identified bamboos planted, and we are monitoring regularly. And in that, most of the plants are seed source. We regularly travel to different countries to, if we came to know that seeds are available, we immediately go there and we collect the seeds there and we bring back to India and we check whether that is performing good, whether that is that variety we identify. And we called many scientists in this bamboo industry to check whether these varieties really proper. From these species, whatever we planted near our company, so once we take the explants from this, the performance is very, very, very good, because we take the explants and we inoculate within 20 to 30 minutes after the treatment. So the performance is very, very high. If we do this, the tannins, what we call the blackening, will reduce automatically in the initiation time and the multi time. So this is the main thing. So always collect the explants and immediately take into the lab immediately. So as we know, the tissue culture technique is already, so we collect the explants in bamboo, we collect the nodal thing, then we sterilize it with multiple disinfectants. So it's based upon the variety and based upon the which time we are collecting. So we initially followed the basic standards for everything, but in the final we came to know there is no standards. So we have to play accordingly as per the cultures. So then we do the initiation. In the fourth image you can see. So this is the initiation stage. And this is our tissue culture facility. So this is how we inoculate. The inoculate video, it will be played after our PPT. There is one more thing is that we should only take 1 or 2 cultures from the bottle. We should initiate and transfer immediately to the bottle. Such a way it will reduce the tannins formation inside. The time, how much minutes it comes out and how much minutes it goes into the new bottle, it matters everywhere. This is the initiation stage. This is we call it as a transfer stage. From the initiation, we get the cultures like this. Then this is the multiplication stage. This is especially a tulda, bambusa tulda. It is in the second stage of multiplication. And this is the final rooting stage. So you can see the different things, how from initiation to the rooting comes. It is from a different thing. The first is the Newtons, and the fourth bottle is the Dendrocalamus asper, and the fifth is the Stocksai, and the sixth is the Newtons. And the growth room, it is a very important factor. So whatever we do at inside the laminar of chamber, so we should maintain a proper climate and the atmospheric condition into the growth room. When we talk about banana, we usually follow a common, very, very common thing, degrees and common temperature, common lux and lighting timing. But in the case of bamboo, each variety is having each timing. So we had so much of research, so much of trial and error, and we standardized a protocol. We usually maintain for bamboo, for the multiplication, from initiation to multiplication stage, 20 to 24 degrees Celsius. For the rooting, we maintain from 25 to 27 degrees Celsius. If you go beyond 27 degrees Celsius, there will be blackening of the culture. And before the rooting forms, the culture will become drier. And also, for the second we usually, the people transfer after 3 or 4 weeks from one bottle to another bottle. But we recommend to go for the rooting stage before second week. So in the second week to third week time, it is better to transfer, then you will get the good rooting. And the lighting lux, we give 1,500 to 3,000 lux. And also it depends upon the variety, we change accordingly. And the lighting timing is 4 to 8 hours. We usually give around 5 to 8 hours for this multi, and for the rooting we give less hours, because when the less hours there is a stress happens, then the rooting will be high. And these are the primary hardening plants of bamboo. This is the left side, the image is the washed hexagar asper. And this is a little primary hardened plant of Dendrocalamus asper. And we use 100% sterilized cocoa pit. And also we use RO water only. So we have a RO unit around 40,000 liters per day we are producing. Whether it is a polybag, secondary hardening plant or primary hardening plant, we use only RO plants. Such a way we can reduce the salt attack. If you see the plants, it will be in a good color, green color up to 2 or 3 months. Then it will be slowly becoming yellow. But if you use the good water, even 6-7 months also, it will be good. We keep in the plants that are around 30 to 45 days. And again, it depends upon the plant varieties like Oliveri stock, say it will take more time since it is slow growth. And for bamboo, we maintain around 8,000 to 15,000 depending on the variety. Lux. And humidity, we maintain 50 to 70%. Again, it depends on the variety. Then we come about secondary hardening. We use 30% red soil, cocoa peat 50%, 20% sand. We can increase the red soil concentration as well. We are increasing cocoa peat because to reduce the transportation cost, because we ship all over India. So the transport will be very high. And the water TDS, we are giving less than 50. And after 2 months of planting, plants are usually ready for sale. And in this case, what we are doing is, so some people in Delhi, some people in Uttarakhand, they will ask for 1,000, 2,000 plants from us. So we cannot ship from Bangalore, Hosur to them, no? So what we do is we introduced a different condition called a paper pot. We call helipods. We have an imported machine from Denmark. So that will weigh you less than 50 grams. So that we are couriering it and we are sending through the flight. So that will be shown on the video. And again, the main important factor you should consider is the main water. TDS is highly important, whether it is a polybag or as a primary hardening plant. It will perform the plant's growth well, and even the mortality you can achieve up to 95 to 98%. But most of the people in this industry, they are using above 200 TDS, and if in Hosur, if you tell, it is more than 500, 600. So the water is the main factor, and after using this good water, our the fertigation cost is reduced drastically. And these are the basic bamboo plantation guidelines we standardized. So we recommend around 5 to 10 feet the planting distance, approximately 850 plants a acre, and 5 into 12 feet if we plant is around 750 plants a acre. And depending upon the soil, it grows everywhere in whatever soil except sand. It grows everywhere. The only thing is that the growth will be slow. And the harvesting time 3 to 4 years. For some varieties, it goes up to 5 years, like Dendrocalamus asper. We are expecting poles per plant per year from the third year is only 8 to 12 clumps every year. At the good maintenance in our company, we had up to 18 poles a year per plant. And each pole will weigh you 10 to 15 kg. So this will increase as the age increases accordingly. And up to the water TDS is 0 to 2000 to 3000 ppm we have tried, because in our company, the water quality is around 1600 TDS. So in the TDS itself, it has grown up very well, the outside plants. And we also supply to a company in Gujarat. They are irrigating the effluents there. So still the bamboo plants are growing up well. So if you're considering 850 plants a acre at good precision farming, we can get up to 80 tons, but people usually get 30 to 40 tons regularly. And if you just sell it as a firewood, it is around 4 rupees a kg. It will give you from 80,000 to 2 lakh rupees just as a firewood. And now many of the cement companies, came back to this bamboo because they need a huge coal or fuel to burn the cements. Usually they use for 1 ton of producing a cement, they use around 300 kgs of wood. So these bamboos, they are planting and they are using on their own. So then the next thing is that now the climate change is the biggest factor. So we know already the bamboo is the fastest carbon sink, and the normal forest, it will sink up to 18 tons of carbon per year per acre, whereas the bamboo, it can sequence up to 60 to 70 tons of carbon per year per acre, which is 3 times higher. So since the carbon is 3 times higher, of course, the oxygen is going to be 3 times higher. There is an oximeter test we did at our company campus. So in our bamboo setup, we had oxygen level up to 30 to 35%, whereas in the average year, it is having around 21% of oxygen. In the cities, in the Osur, it is less than 15. If you go deeper, Chennai, Bangalore, it will be less than 10. And I want to tell something about, because we want to talk about the application as well. So where it can be hugely usable for the farmers. So I'm deeply talking with many sugarcane people, the retired directors of the sugarcane people. I got the information from this. The sugarcane has to be planted every year, whereas the bamboo, it is not like that. Some variety, it is a lifetime crop. Some variety can be planted 30-35 years once. And the water requirement is very high for sugarcane, at least 2 to 3 days once they have to water it. And for the bamboo, mostly the people never water. So initially they water and they leave as the rain-fed. And for the sugarcane, huge labor and more fertilizer required. For the bamboo, less labor and mostly people never fertigate it. So technically, in 1 ton of sugarcane, they are taking around 10 liters of ethanol. In 1 ton of sugarcane, only 10 liters of ethanol they are getting produced, whereas in bamboo, it is up to 340 liters of ethanol is getting produced in 1 ton. Whereas in sugarcane, the yield per acre is 40 to 50 tons. And in bamboo, we can calculate as a 40 to 60 tons, but it goes up to 80. We can calculate average. The ethanol produced in 1 acre through sugarcane is 400 to 500 liters, whereas the ethanol produced in bamboo in 1 acre is 10,200 to 12,000 liters. So recently, the Indian Oil also they have accepted to buy the ethanol at rupees 59. I read an article in Times Now. It is in Maharashtra, the Maharashtra Development Board. So they told us. So the ethanol is around rupees 59 per liter. Approximately, if we produce the ethanol through the sugarcane, it will be 23,000 to 30,000 per acre. Whereas if we do in bamboo, it will be 6 lakhs to 7 lakhs per acre. It is not a simple thing, because the sugarcane, we are only using it as a food and as for producing power. But bamboo is not like that. From the furniture, from fiber, from electricity, bioethanol, and as poles or for biomass. So everywhere we are using it bamboo. So the applications are huge. When we talk about the people selling it as a pole, we calculated 8 to 12 new clumps per year per plant. It is coming at medium maintenance. So each pole is weighing 10 to 15 kg at the 3rd year, and it will increase up to 25 kg from the 5th year. Approximately, we get around 7,500 poles per acre, calculating 750 plants, giving 10 poles a year. So here, the least pole rate is around rupees 40, and the highest we have seen is 300 based upon the dia, thickness, input distance, and variety. If we calculate the least price is, if we sell at rupees 40, we get up to 3 lakhs per acre per annum, and around, if we calculate 300 rupees, it is around 20 lakhs per acre per annum. And the application of bamboo plants is that we know that it is a huge into biomass electricity, sugarcane industry. So now we are talking about women employment. So before the corporate companies came into the factor, only the rural ministry has promoted more number of agarbatti, matchbox. So they give these bamboo sticks, agarbatti sticks to different villages. So they made the coatings of the agarbatti, so they collected that. Still, this is happening at many northeast area and deep villages. So now the corporates came into the factor. They completely took the entire part into their industry. So the women employment has came down. The rural employment has came down. For agarbatti and for handicrafts, so this will be really useful for Ministry of Rural Employment. Now, I have seen a textile industry at Thailand. They are producing bamboo, cotton, and it is already available everywhere in the Amazon, Flipkart, and it is antimicrobial. Paper industry, they are doing it. Bioethanol, so recently in Assam it has started, and even in Hyderabad, they are planning to start 40,000 liters a day through bioethanol in Andhra Pradesh. And furnitures are there, food industry, carbon credit is the important factor. Last month, a person from Singapore came to us and he was telling about carbon credits, but in India, it is not still happening. We request the support from the government, because the countries like Singapore and Dubai, they don't have good soil and there's no proper land. So if India promotes this, it will be very good. And also we planted some of the bamboo plants at the Nepal government to prevent landslides at Himalaya from the Kathmandu to Chitwan area, little areas. And also planting along road medians is also good. So this is my basic explanation about bamboo through tissue culture. Thank you so much.
Thank you, Mr. Gautam, for this very comprehensive presentation. And it was very interesting. Though I discussed this culture throughout the day, but I found this very, very informative. You talked about this culture as application in paper, textile, bioethanol, and many more. So thanks for this nice presentation.
Thank you.
So now we'll move to the next presenter. And that will be again very interesting, the teak tissue culture. And that will be presented by CEO of Mother Biotech, Dr. Balakrishna Muthukari. He is a founder, scientist, and the chairman of this company. So you're not the CEO, chairman. And a vast experience in teak tissue culture. So just I think I have missed something. Before we move to the Dr. Muthukari presentation, we'll have a brief video. Mr. Suez, can you show that video? Let's from your side. Bamboo tissue culture, a brief video. Thank you. This video gives feeling of personally visiting any tissue culture facility in commercial. So thank you. So as before this video, I was discussing about Dr. Balakrishna. He's a young entrepreneur doing excellent work in teak tissue culture. And he has more than 26 years of experience in tissue culture. So I think he has been doing teak tissue culture from the early childhood, it seems. So he has many publications. He's a consultant to Andhra Pradesh Forest Department, and he's exporting plant to many countries. So he also published book on micro propagation techniques of tree species. So Dr. Balakrishna, over to you now. So I think my colleague will project slide from this side. Mr. Suez, can you share the slide now?
Hello, this is Balakrishna Muthukuri, founder of Mother Biotech. Little did I know at the age of 15 years that an innocent challenge with my father would land me here today. I thank to BCIL for giving me an opportunity to participate in this webinar to give my inputs on this tissue culture teak. Teak is known as the king of timber and is high demand due to its natural properties like resistance to water, pests, rats. Plus, it has minimal shrinkage. It is not corrosive when used with steel and is highly durable. Due to these properties, it is being used in making furniture, doors, bridges, boards, and ships since ancient times. And its propagation was through seeds. The wait period for seed germination was nearly 2 years due to its hard outer coat. After planting the seedlings, farmers had to wait for many years as the yield would take anywhere between 25 to 70 years, which is known as the next generation crop. Despite waiting for such long periods, the wood volume would be less because there would be a lot of wastage due to irregular growth. Over a period of time, when the demand for teak was increasing and percentage of natural germination and of seeds was declining, cultivators innovated methods and treatments for artificial germination, propagation of root systems to get ready to plant seedlings with a shorter period. But they also started silvicultural practices like manuring, pruning, a treatment for diseases, and other related works to ensure that there would be better growth and minimal wastage. However, the time consumed for harvesting remained the same. As on date, the natural germination of seed is almost negligible. There are so many cultivators of teak in all the tropical countries, and teak is the best agriculture commercial crop till date due to minimal maintenance and huge returns. My inclination towards teak started when I was in high school. I used to accompany my father, worker for AP Forest Development Corporation, to his propagation center whenever I had a chance and learned various methods of macro propagation of different plants like budding, grafting, clonal propagation, vegetative propagation. And he was a gold medalist in eucalyptus propagation and would give me a lot of tips for successful macro propagation. He briefed me about micro propagation and said that the same was impossible in teak. I challenged him that I will produce micro propagated clones, which he rubbished with a smile, but I did after 15 years. I started studying every information I could get on teak from libraries, department, and started my preparation for initiating the propagation. I selected few teak trees based on different parameters from different agroclimatic conditions and started working on them. But it was a tough journey and took almost 8 years for me to initiate the bud and took almost 2.5 years from then to successfully get a aseptic plant left because of the fungal contamination, bacterial contamination. I could successfully develop 9 best clones which are suitable for almost all the 9 agroclimatic conditions of tropical conditions. We are planning to increase up to 20 clones after 5 years. These clones will give you very good yield in a short span of 8 years, just like their mother plants. This product is a revolution in agroforestry.
Sir, you are muted.
Yeah, our trees can be grown almost along with the seasonal rotational crops of a particular area to get the benefit of immediate cash returns. For livelihood, along with the long-term cash returns to elevate an individual economic status. Teak is also the best to reduce global warming as it has maximum carbon sequestration when compared to any other crop, any other tree. This was also stated by GER, India, after they conducted research on different trees and they stated that by the time a tree attains a girth of around 10 to 30 centimeter, it have consumed around 3.7 lakh tons of carbon dioxide. Hence, I requesting industries and all the industries responsible for CO2 emissions can go for teak plantations to compensate on the carbon footprints and to reduce global warming. My focus is to give the best clones to the world and create a revolution in changing the economic status of the farmers. I have already started the journey by giving clones which require minimal maintenance and silvicultural practices. And it will continue in search of better trees and planning to propagate to produce more than 25 clones. Next. Sir, Mr. Shukla, sir, can you show my PPT? From the beginning?
Yet is there. We are on the return on the investment.
Yeah, from the beginning, can you share my PPT?
Yeah, Suresh, can you start from the beginning? Just share the first slide.
The first slide, yeah. Yeah.
Can you see that, Balakrishna?
Yeah. Our mother biotech is world's exclusive producer of high-yielding tissue culture teak, which are disease resistant. We are producing till now. We are producing 9 best clones. So next slide. To prevent enjoyment teak plants. Yeah, producing 9 best clones of teak based on rainfall, differ from 500 millimeter to 2000 millimeter rainfall. We have customers in 25 districts of Karnataka and 25 states in India and more than 5 countries. This is out of 198 countries, teak plant will grow almost 150 to 120 countries, which we market here. Yeah. Next slide, sir. Yeah, teak wood plays a major role in the timber industry. It will have high demand as compared to other wood. It is not alternative source to other wood. It is king of timber in the domestic and international market. In general, according to my experience, Indian climate and soils are naturally suitable. If you observe, you can see the only 3 varieties of teak in the world. First one is Tectona hamiltoniana. It is endemic to Burma. It will grow in Burma only and nearby places. Next one is Tectona philippiana. It's commonly called Philippine teak. It will grow in Philippine country only. Third one is Tectona grandis, which will grow in naturally in 7 countries, and by planted forestry, it will grow in 110 countries. If it grows in Nelumbur teak, it grows in Balar teak, it grows in
Dr. Balakrishna, we are not able to hear you. I guess there is some network problem at his end. Yeah, please unmute Dr. Balakrishna. There was some problem from your side, network problem. You rejoin again.
Yes, sir. Yeah. Wastage is very 15 to 20%. Even if we have harvested this one, this TCT, you can see wastage percentage in the stumps more because of the irregular growth in stump raised plantations. Our plant can be planted around the year, but the growth rate differs from month to month. In January, a little slow. In March, 1 foot per month. In April and May, 2 feet per month. In July, 2.5 feet per month. In the month of November, only 20 centimeters per month. It's a global analysis report. It is the best investment option when compared to all other, related share market. It is given by US market. Peak versus traditional investment. If you observe, you can. Why tissue culture is invest in tissue culture tea plant is environmentally conscious, long term and stable investment, low maintenance and huge returns. You can claim carbon credits because of this high carbon sequestration capacity of tea plant. We have sent our plans to IFGTB Coimbatore also regarding this carbon sequestration levels. Plantation, if we plant 440 plants per acre at the spacing of 10 feet by 10 feet, you can get minimum 25 CFT of wood per plant from 8-year-old tissue culture tea plantation supplied by Mother Biotech. You can get 11,000 CFT of wood. If you want to continue that plantation, after 12 years, each plant will give you 75 CFT. After 17 years, 150 CFT. This is all proven record. We have a proven record also. This is our return on investment. We observe whatever I have told just now. This is return on investment. Yes. Yeah, I have taken this information from NABARD. Why other crops differ from teak differ from other crops? Return on investment I have mentioned here. Stump teak, seed-based teak, Acacia, Performance, Poplar, Jatropha, like that. This information I have taken from NABARD bank bull projects. Yeah. When compared to all the other crops, according to my observation, teak is the best option for investment. Yeah. Yeah. Because of this high carbon sequestration capacity, the information research done by GER Ahmedabad, teak plant is having high carbon sequestration capacity. That's why I'm asking each and every university to go through teak plant and make some teak plantations to reduce global warming as a corporate social responsibility. Teak health and wealth source. Carbon credits are a vital component of national and international trading. Strategies have been stressed to lessen global warming. Teak has the highest capacity of carbon sequestration. These credits can be used to finance carbon reduction schemes between trading partners. In Uganda also, in Tanzania also, they are working. Most of the companies are working on carbon credits. Next step. This is carbon credit cycle. You can see how the flow. Next. Yeah, this is GER Ahmedabad report regarding carbon sequestration levels, Sectona grandis. You can download from the net also. Stages of tissue culture. In our Mother Biotech laboratory, a total 6 stages involved in the production of tissue culture tea. First one is initiation. It will take more than minimum 5 years to 10 years to get aseptic plantlets. Why? Because you can see 90% initial stages, you can see 90 to 95% contamination, bacterial fungal contamination. When we take expand from the mother plant, is the major problem in teak plant. I don't know what will happen in other species, but in teak, it is very difficult to find, to reach that aseptic plantlet level. It will take minimum 7 to 9 years. Even a layman also can identify the tissue culturized plants and root plants. You can see a continuation in the shoot with the root, the left side, and the shoot grows to one side from the root. This is a stem raised plant. You can easily identify this. If you observe and you break one plant, you can easily identify this. This is our mother plant. This is 17 years to 18 years old, almost 135 feet height. It is in Bhadrachalam, APN BV, Andhra Pradesh, North Bhadrachalam and Venkatapuram. Their rainfall is 700 to 800 millimeter. We named it Mother MBT 700 clone. MBT Mother Biotech teak 700 clone. If you plant this within 17 years, it will grow up to 120 feet and 175 centimeter girth. It is one of our mother plants. Yeah, this is the initiation of teak plant. You can see, you can observe this. This is after 2 months of initiation. We have to cut. We have to shift to other fresh media more than 25 to 30 times to get one aseptic plantlet length of 1 centimeter. Like that, it is very tough critical stage and tough stage in the production of tissue culture teak. Yeah, after 2 years from that initial explant, after 2 years, we got like this. Totally this protocol differs from multiplication protocol. This is a 1.5 year old plant, aseptic plant. From here, we have to standardize mass multiplication media, mass multiplication protocol. This initiation protocol differs from mass multiplication protocol. Yeah, this is mass multiplication is going on, inoculation, mass multiplication, and this is a prime rooting stage. In this tub, we are using the SV rooting of teak plants. This is rooting stage. We can create microclimate here to get roots. Here we will maintain 90% humidity and 30 degrees centigrade to get advantageous roots. Once this rooting is done, later it will be sent for primary hardening purpose. This is rooting, 99% success rate, if you observe. Once rooting is done, we will transfer it to poly bags. There will maintain 20 days to get 12 inches plant. This is our primary hardened center. And after that, this is the transport. This photo is when I work for government, AP government, we have transferred to Ahmedabad. These are the stages. First photo, this is mother plant. Second one is explant. Third one is preparation of media and inoculation, mass multiplication, rooting in the tubs. This is advanced root formation in the micro-propagated plantlets. Next, primary hardening, secondary hardening. Those plants are ready for planting. And this is our 8-year-old plantation in Andhra Pradesh. This is 2 months old after planting. This month of planting. This is chrysanthemum variety. In Karnataka, one of our customers planted this chrysanthemum variety, highly economic value. This is our 1-year-old. This is our 1-year-old near in between Karnataka and Andhra Pradesh. This is our 1.5-year-old plantation. Yeah, this is our 9 to 10 years old plantation. We harvested this. We observed 84 centimeter there. And this photo is taken by Korean ambassador when he visited to plantation. He has taken these photos. Above one, our 8-year-old plantation, 100 centimeter girth. 95% of our customers are satisfied with the plant's growth. You can see 70 to 80% hardwood formation. Here, our plantations, we have to add some more. We have plantations in almost 20 to 25 states of India and 25 districts in Karnataka and 5 countries in the world. Uganda, Tanzania, Cambodia, we have plantations. So this is our 9 years old plantation. He is a South Korean ambassador. He has visited and he has taken photos and he has forwarded to me. You can see 80% hardwood formation. in tissue culture plants.
Dr. Balakrishna, sorry to intervene. Your presentation is very interesting, but we are little exceeding the timeline. Can you just please conclude?
Yeah. Oh, it's over, sir. It's over.
Or
Yeah, once again, thanks to BCIL for giving me this opportunity to participate in this webinar to give inputs on TEAG. Thank you.
thank you, Dr. Balakrishna.
Yeah,
And this is very important topic. The TEAG tissue culture is not so easy, and we have received so many queries from the audience.
yeah, that's right. Yeah, thank you, thank you.
You have been wonderful work. So whatever query we'll receive today and later also will connect to you that how to resolve those queries, technical queries regarding the TEAG tissue culture. So we also have a brief video on the TEAG tissue culture. So just project immediately. 1.5 minute video. So that will give idea about the process of the TEAG tissue culture. Yeah, Mr. Suez, are you playing the video?
Dr. Shukla, if it is a problem, then to utilize the time because we are running late, you can start the next item.
Yeah, I think there is some technical problem in that video. So I think before we move ahead for the panel discussion, which is very important, just will have a brief poll with the audience. So Mr. Sarath, this project poll. So it is a brief poll as we had done the first time. This is the second one. And again, there will be 3 questions. We have projected this poll in front of the audience. And this is clearly 3 questions. That first one, do you agree that tissue culture of tree woody plant has the potential to improve the forest cover and accelerate the growth of timber-based industry at the national level? So you have to select either yes or no or not sure. Second one, are the quality management practices beneficial for improvement in the production process of superior tissue culture plant of tree or woody plant? Yes or no, not sure. 3 options again you will get. And the third one, do you agree that micro propagation is the efficient methodology for the production of tree woody plant on a commercial scale? Just 3 options you have for each question. Yes, no, or not sure. So we are getting response from all the participants. We'll just wait for 5 seconds and we'll close this poll. 5, 4, 3, 2. Now we can stop now. Just show the poll result. Okay. So 97% agree that it has potential to improve the forest cover. So it's a very prominent technology and it has promoted in a big way. So that is a message from the audience. And the quality management practices is highly beneficial. At 94%, they told that it is important for production of superior plant. And again, 91% they have told that they have agreed that this technology is highly efficient for production of woody plant at commercial level. So this is very important finding. And as an organizer, we have taken note of that. So thank you so much. Please stop the poll result. So if the video is ready of tick tissue culture, I request my colleague to project that video in a few seconds. If it is feasible, just try once, because this will give idea of the scholar student, those who are not visited commercial facility, they'll get good idea about the process. Okay, so there is some difficulty. We will not be able to project that video. In fact, we'll combine this YouTube video, and those who would like to see this, they can see later on. So with this, we'll move to the very important segment, that is a panel discussion. And the panel discussion, we have eminent expert with us today, Dr. K. Gurumurthy, Dr. Chandrasekhar Biradar, and Mr. Abhay Gandhe. I'll briefly introduce without taking much time, because already we are running behind schedule. Dr. Gurumurthy is very key person in this area, and he is the former director of Institute of Forest Genomics and Tree Breeding. So he has extensive experience in different capacity. I'll not be able to tell all those things, but he also helped us in setting up certification program and institutes in the country. And as a head of IFGTP, he has taken various initiatives and participated in various national and the international committee in the area of tree biotechnology particularly. Dr. Chandrasekhar Biradar is important person at global platform. Is a country director of Center for International Forestry Research, C4, and World Forestry, and also chief of party for TOFI, that is called Tree for Outside Forest in India. He is involved in outreach activity, ecosystem, and he is promoting various kind of research which is leading to the rural livelihood. He has worked in various capacity in last 20 years, like researcher, principal scientist, manager of various research units, and he has published more than 400 papers. So he's a very important person, Dr. Chandrasekhar Biradar. And now third panelist, Mr. Abhay Gandhe. Mr. Abhay Gandhe is the senior independent agriculture consultant and working for the rural livelihood. He has very rich experience of more than 38 years in managing various key programs in India and across the country. And he has been working with the Tata, NABARD, and particularly for the bamboo and tree species like apricot, cashew, mango. So currently he is a freelancer consultant. So with you see you have the diverse range of panelists, and our participant would be greatly benefited. I hope so that they will greatly benefited with this discussion. So in this panel discussion, just briefly, I will flag few key questions that has been received from the audience, and they would like to know from our eminent panelists that what are the scope, prospect, and impact of this entire exercise on tree culture. So first, I will go to Dr. K. Gurumurthy, former director, IFGP Coimbatore. So Gurumurthy sir, my question for you is that in tree tissue culture, often TC-raised plants are plegiotrophic because of the maturation, and selecting the juvenile tissue is very important. So we'd like to know how to select the juvenile tissue to start the tissue culture process. Gurumurthy sir, over to you.
Okay, thank you for giving me the opportunity to be a member of the panelists. Juvenile, it is very important to select juvenile tissues because the trees pass through maturation phenomenon and they become plagiotropic if you select orthotropic shoots which are chronologically young but ontogenically much older. Therefore, you have to select tissue from either epicormic shoots, which are shoots buried in the main stem, or the shoots that arise from the cambium at the bottom of the tree. There are many ways of developing juvenile strategies, and one of the strategy that is often followed in eucalyptus is cut back the tree
and then it produces epicormic shoots, which are all very young. Or it is also possible to take out the cambium, small amount of cambium at the bottom of the tree, and then culture it, produce shoots, and then convert it to multiple shoots quickly. This is one way of doing it. Different people use different strategies, but one should see that there is no plagiotropism as far as tree species are concerned. Otherwise, the whole purpose will be defeated. How to select juvenile tissue or juvenile shoots is the prerogative of the tissue culture specialist. That has to be looked into. Or you can even use what are known as the shoots that come out of the root zones. If you dig up, in case of sandal, very often you will get these shoots, and these shoots are also juvenile. There are many methods of selecting juvenile shoots so that the cost is very effective. Thank you.
Thank you so much, sir. I think this will give insight those who are willing to propagate tree. So I would go to now Dr. Chandrasekhar Biradar. And as I told that he has been handling many activities across the globe. So Dr. Chandrasekhar, your video is off. You are also muted. So till the time you get your network connection proper, I would go to Mr. Abhay. Mr. Abhay, you have been doing lots of work at the ground level by doing various socio-economic work at ground level, particularly bamboo and tree species. So regarding the bamboo as a topic of this theme program is bamboo in a tea, I would ask you, how can the bamboo tissue culture supply system it will create a scope for rural livelihood of rural women? How you can connect this tissue culture supply system to the empowering the rural women?
Yeah, thank you, Dr. Shukla. So my inputs will be coming from this background of mine, wherein I have worked extensively in transferring the technology of bamboo to the rural women through their programs. As in the morning, Mr. Charanjit Singh was saying that now the department is trying to design a program of reaching out to 1 million of households through this bamboo program. And the input on this would be valuable for such kind of large programs. Now, when we compare, say, a tissue culture plant of bamboo and a tissue culture plant of banana, their end use and the end location of plantation of those two species is completely different. Banana is generally planted in a very good soil under very best type of management conditions. If bamboo has to be taken to the mass scale, is to be assumed and is a practical reality that it may be planted in quite suboptimal conditions. So what's the solution of ensuring a proper delivery as well as establishment of bamboo on a mass scale with the rural households? Could be a requirement, this is a clear suggestion from me, could be a requirement that there has to be a third layer of hardening after the second hardening. And this third layer should be in the field nursery, in the conventional nursery, where the bamboo from the second hardening may be kept for at least 6 months till it forms a strong bulb at a strong rhizome at the bottom. And then this plant can be taken for plantation. Now, the rural women through their SHG network, which is quite an extensive network built through the Government of India program, those women can play a very critical role in ensuring this third layer of hardening for bamboo plants. And this automatically should empower them economically also, because a part of the selling price of this third hardened plant would be shared with those women entrepreneurs. So I see a distinct scope of developing a series of women entrepreneurs in rural areas for this one particular requirement of a third layer of hardening of bamboo. Okay, thank you.
Thank you, Mr. Abhay. It provides very good insight how this physical system can be connected to the women empowerment. Now, when we talk about that connectivity with the ground, that market is very important. And marketing, it will make the entire process successful, because unless until it has to be customer, that scalability will not be feasible. So again, I will move to the Dr. Gurumurthy, that regarding the marketing part, then who will buy this tissue culture as tree plant? In other words, how to create the market for tree and woody species. Dr. Gurumurthy sir, over to you. You're muted, sir.
This is a tricky question to answer. Number one, how to create a market for tree species. In case of banana, every farmer will come repeatedly every year, so the market, we know the market, how to predict the market. But in case of trees, or for that matter in case of bamboo, if a person with 10 acres of land comes and buys plants, he is not going to come back to you for other 10 or 15 years. This is a big issue. Now, how to create a market? Is people should think innovatively in terms of assessing the value as related to the land requirement and predictive strategies for the coming 10 years, so that there is some kind of a rotational management of demand. This is very important. How to do that? And will that rotational management of demand will be economical and keeping in view the requirement of plants, land, yield, use of those things. Some kind of a marketing strategist should work on this. They should work back and provide answer to the tissue culture people, so that the demand and supply is well balanced. That's all I can say. But there is no fixed solution for this, like in case of banana or pomegranate, or for that matter, any medicinal plants, which can be grown annually or biannually. Trees have to be grown once in 10, 15 years. And this is an issue that I think tissue culturists need to consider. Maybe some IIM group of people can work out the strategy and provide the marketing solutions for this. Thank you.
Yes, sir. This is a tricky question, and nicely it has been addressed. But as industrialist, like Mr. Palni and Dr. Balakrishna, they will have to work it out how to connect with the market. Now, Dr. Chandrasekhar is there. As I introduced him, that he is playing very key role in promoting not only research but extension work for the tree tissue culture aspect, and he is also contributing policy making, extension work like that. So, Dr. Chandrasekhar, question for you is that what is the need of quality plant culture in tree species? And we'd like to know from you a global perspective.
Yeah. Thank you, Dr. Shukla. I think the very, very interesting discussion happening on this aspect. The one of the we leading called the Tree Outside the Forest India program. And when it comes to the why agroforestry or the tree planting is not become much popular here in India or elsewhere, because of often the first thing is that when we're putting that out, planting the tree, it produces this much and carbon sequestrations, mechanism, all the things there. First farmers are that where I can get that quality planting material. Where, so this is where the need of the quality planting material is not existing there. And a lot of the nurseries we are in place, often the certifications or the quality planting is not ensured. Because a lot of the private sector is also running, and often is not ensured these mechanisms. And this is where we're trying to looking into that how to improve the high-tech nurseries and quality planting material that can provide a perspective for the return of the investment. So because of the trees being a long gestations often, and if you're not quality planting ensured in the beginning itself, and the return of the investment will be in the risk for the many farmers. That's why often they plant mangoes or casuarina, whatever the different species in the agroforest systems, and what they promised, what they expected that's going to happen at the end of the harvesting or the gestation period. I think this is where the quality plating will become one of the key important aspect. And within our all the agroforestry programs and 3 outs of the India, like a TOPI program, we're putting the QPM is one of the major aspects need to be addressed in terms of the not only like the meeting the demand and supply, also the certification standards that the farmers can available, also ensuring that demand and supply gap, connecting the farmers to the nurseries. I think that's the other aspect you're trying to work on that. I think that did I answer your question?
Yeah, Dr. Chandrasekhar, this is very important answer for the very important question. Now, again, I will come to you for the next question.
Sure.
I would request kindly provide some insight on the suitable tissue-culture-based agroforestry system and how it can bring science-based solution towards challenges being faced for the forest landscape. Agroforestry system. What do you, Chandrasekhar?
Okay, so the one of the important aspect into that one, why the quality standards and the insight in the sustainable, this is not available. In fact, there is available. But some of the insight you look into that the suitable tissue culture, the based agroforestry systems in India, and already popularly what we call a more popular popular species into agroforestry systems, whether take it like in the past. The most of these exotic one now the Melia and neem and chandel and rosewood, the established system is in place. But in order to make into the scalable mechanism, still it is not that what we expected there. So this is where I need to be the investment in terms of the incentive mechanisms, in terms of the land holding. That's what we are trying to bring into what they call the high-tech nurseries in establishing the state. So that provide a kind of the standard mechanisms to be taken into the more wider scalable where the private sector can come into place. And also looking into the gaps in terms of the identifying the right species for the right location for the right reason. For example, often the teak become a viable species and everyone started planting the teak everywhere. Even the quality of the soil quality not met for the teak. I think this is where need to be addressed the suitable species for the given locations, and also understand the mechanism for what is the extent of the demand is there in advance. And also often we see when someone want to do the agroforestry or tree planting, they suddenly jump into that one without understanding the existing mechanisms or available planting material. I think these are the key issues need to be addressed, and this is where most of the sectors are moving that side. And I think you asked the second question. Can you repeat the second one, the part of the question?
No, this is your all questions. The entire concern, what you explained that this is all the concern queries of organizer as well as audience. So because we have, thank you so much, Dr. Chandrasekhar, and.
I would like to add a bit on that one, like in the 3 outs of the Forest India program, and we're trying to bring to the science-based the solutions. One thing we're trying to removing the existing bottlenecks in terms of the policy and some certifications and the intensive mechanism. Third is also whether we can bring it to the insurance scheme into that the tree base, because of crop systems is well established mechanism in place. Whether it came the subsidies, whether it came to the MSPs, return of investment, but that's not in place into the tree-based systems. So this is where we're trying to bring it to the landscape of the forest landscape.
Yeah, right. The science-based solution is very important. And this discussion is very interesting, and it may continue throughout the day, but since we have the time limitation, and also we have received large number of queries from the participants. So with the permission of esteemed panelists, can I move ahead for opening the floor for the question answer? That will add more value to the program because we receive large number of questions. So I would like to start this panel discussion, if you permit me.
Sure.
Yeah, Mr. Abhay, is it fine?
It's fine, sir. Please go ahead. Absolutely.
Okay. So before we move for the open discussion, just like to inform that we'll have enough time for the discussion, and we got a query from participants that will project it one by one. and I request all the panelists expert to be available to address their queries. So before we move ahead for open discussion with the participants, I request my colleague to project the poll, third poll. It's also very important, so that we'll get the feel of audience what they are thinking about the program. This like a feedback. The third set of question that how do you rate this webinar in terms of the enhancing your knowledge with reference to the technique for the production of tissue culture of tree woody plants. So this feedback is very important for us. I request all participants to respond on that. And the second question, would you integrate knowledge gained during this webinar in your research or extension program? So yes, no, not sure, you need to select anyone. And the third question, does this webinar encourage you to popularize tissue culture technique and establish tissue culture facilities in your region or country? So please respond to this question. I request all the participants, still the large number of participants, those who joined from the beginning, they are with us. So just 5 more seconds, please quickly respond. And just to like to announce that we will provide certificate to all the participants active during the entire session post webinar. So last 5 seconds, 5, 4, 3, 2, 1. So please show us the result. Okay. 26%, 56, excellent. 34%, very good, like that, good. Some 2%, there's fair. Okay, that feedback is very important. Now, 94% they told that they would like to integrate this knowledge for their research and extension program. That is very good insight. And 96% they told that this program encourages them to popularize tissue culture technique in their countries. So thank you so much for participating in this poll. So now I think Dr. Tyagi, we can open this forum for the discussion. And I have the print of those queries received from the participants. So Dr. Tyagi, I would like to proceed. Are you going to take up those queries and I'll supplement?
Yeah, thank you, Dr. Shukla, and thank you all the speakers and panelists. In fact, this has generated so much interest from the participants, and I see number of questions, almost 40, 50 questions I am seeing on the windows in the question answer, which of course some are repeating. But Dr. Shukla, it is not possible to answer all the questions.
Okay.
I may pick up very, very typical questions where it can be answered, but about the medium for the contamination control, phenolic controls, this I will leave it because this cannot be answered in such a short period. However, we would try to assure the participants, we would try that as many as questions which are not answered, we will contact later to the experts and try to send the information to the participants. So most of the general questions I will be taking up. The one is what are the problem associated with commercial production of tissue culture plant for economically important forest species or woody plant species. And who would like to respond to this basic question, though important? Maybe Dr. Gurumurthy?
Dr. Balakrishna wants to respond on that. So
Okay.
Regarding commercial production of woody plants, major constraint is contamination. At the time of surface sterilization also, we have to sterilize suitable chemicals, I mean antioxidants and media also. Regarding teak, it is very difficult to initiate one bird you can see 90% problems in surface sterilization of teak bud. To avoid this contamination, I have standardized on protocol. Flow also, process also. First, we have to wash the explants under running tap water for 10 times. Next, we have to keep these explants, juvenile explants, in TPAL, 2% TPAL solution for 10 minutes. Next, mercury chloride. Finally, in 70% ethyl alcohol. These surface sterilization techniques regarding teak, I have standardized more than 4 surface sterilization techniques. In all techniques, I am getting 90 to 95% bacterial, fungal-free explants. It differs from plant to plant. If anybody wants to know this surface sterilization technique, I am ready to share my experience.
Okay, we'll connect such query to you directly later on.
Yeah.
So there are some questions which are of very general. I am leaving them. And the questions which have been raised to contact for the expert, I think the participants are requested to contact Dr. Shukla. He can connect you with the expert for those questions. The one question I would like to take that Raju Karthik has mentioned that I have some doubts. How about RITA bioreactor, RITA bioreactor? Is it feasible for commercial production?
Feasible, but in teak it's not required. If you apply this, we'll get more source.
Okay,
It
thank
differs
you.
from plant to plant.
It varies plant to
Okay.
plant.
Yeah, it differs from plant to plant. In ornamental plant, tissue culture plant propagation, some tissue culture laboratories are there using bioreactors to get
Okay,
more
thank you.
and more yield.
Thank you, thank you.
In UD plants, it's not required.
Okay. So one question is from participants Camilo from Mozambique that I am trying to introduce bamboo in tissue culture, but having a lot of contamination problem. So I think tissue culture is facing this problem at initial stage, but you'll have to standardize and you'll have to go through the literature because Dr. Sushmita has covered the comprehensive protocols. Maybe you can consult them and to work it out. The another question is that whether instead of RO, instead of sterilized water, this RO water is used for commercial production. Tap water is used for commercial production instead of RO.
Oh, yes. Actually, the tap water can be used. There is no problem if the TDS is low. So if the TDS is high, they should go for the RO water only. There is no option. So they can have RO water which is below 100 TDS or 50 TDS. We are not recommending below 20 TDS. So accordingly, they can use the filters. Because why we are telling not to put more TDS? Because the salt attack will happen and it will make the plant to not absorb the nutrients, and slowly the pH of the medium will increase and it will die slowly. So it's always better to use low TDS water, whether it is a tap water or RO water.
Okay, thank you. Rosa looks like from Philippines. She would like to know the culture media for the micropropagation of bamboo. So from industry person or the Dr. Sushmita has covered some culture media. Rosa, you are advised to contact them and they can give you more details later stage. Another question is that what is the most popular variety of bamboo in India for commercial purpose?
Especially it is depending on the application only. So if they are looking for biomass purpose or for firewood purpose, they will go with the varieties like Balkova. If they want to go with Agarbatti, means they have to go with Newtons and Tulda. If they want to go for multiple like construction purpose, they should go with Asper, Brandisei, then Newtons and Tulda. So it is depending upon the variety. So for edible, they have to go with the Asper and Brandisei. So it's depending upon the variety, we can recommend them. So what purpose they want to plant the plant, we can help you out to that.
Thank you so much. Dr. Shukla, one question is for you would you be able to share the presentation and all videos to the participants?
Yeah, video, it is live on YouTube, but again, we'll share the link so that they can see all the recording.
Okay, thank you.
And one person asked, when is the program for the ornamental? So the 26th August, that is the final program of this series. We'll have this program for the ornamental tissue cultures on 26th August. Please visit BCL and Apari website for the details.
So one question is, sometimes callus comes and shoot while multiplying the teak. I think a general question where I can put forth the possible reply that because it depends upon the medium, and if you are using more auxins, then the callus formation is there, but you have to slice variety to variety or the plant to plant the medium. So this is a matter of research and what variety you are dealing with. Another question is, which part should be taken as explant for teak propagation, and how many weeks or months it will take to become successful explant for further propagation? Bhavana Chaudhary asked this question, and maybe Dr. Balakrishna.
Yeah. In mother biotech, we are producing 9 clones. That incubation period differs from clone to clone also. One will take 50 days, the second one 6 months, the third one 90 days. It depends on. Multiplication ratio in mother biotech is 1:10 ratio.
Okay.
Even clone to clone also, it will take 7 to 11 months. One clone will produce within 7 months. Another clone 9 months. One clone will take 11 months. Based on rainfall also, this 6 days period differs from clone to clone.
Okay, thank you.
Yeah.
There are a series of questions from Mr. Khokar, Muhammad Ilyas Khokar, but they are the general question about the tree tissue culture. I would refrain to take because he is asking about the protocols of the in general. So maybe the literature can be consulted for that. But if you have any further question on teak and bamboo, it is a welcome question. Another question is elongation of shoot buds in the woody plant, particularly the teak, if it has been asked by Dr. Professor Tiwari. What are the solutions for elongation of shoot buds in a per unit time?
Yeah. If you use this kinetin and gibberellic acid 4 milligram per liter mid dosage, you will get more shoots and elongation period can be reduced by these 2 chemicals. In general, it will take 50 days. Within 30 days, you will get the elongation chemicals by using kinetin and gibberellic acids.
Okay. So Shalini Purwar, she wants to know the tissue culture. She wants to start tissue culture of mahua. I think Shalini, you contact Dr. Shukla. He will guide you and connect you with the correct person.
So Dr. O.R. Reddy, Dr. Ramalika Reddy, there is very senior expert, and he has asked that how many facilities are recognized under the certification program of Government of India. So 4 to 5 organizations are there, they recognized. So if you also want to speak, so can I allow him for 1 minute?
Yeah.
Key person who helped us BCIL for setting up the NCSTC in the country. Dr. Reddy, if you can, you're active. I have allowed you to talk. Can you unmute and just provide your input? If you can hear me? Yes. Dr. Reddy?
Hello. Hello, could you hear me?
Yes, we can hear you, sir. Welcome in this webinar.
Yeah. It is really very interesting seminar, and this is the first time to take up on the forest species and woody plants, and which contribute not only to the clean environment and also carbon credit. So this should be there should be some governmental priority to support these programs, because in the form of subsidy and also creation of community nurseries, because after primary hardening, secondary hardening, some cases you require a support of further to provide a support through community nurseries. So it involves not only the government, the industry, as well as the organizational support at local level for both extension activity, promoting extension activity, and also to support the availability of the material, tissue culture plant material, because that is the most important, especially when you are dealing with the tree plant species. There is a need for use of some kind of a biostimulants or VAM cultures for getting the plants acclimatized to the environment. Because these are very critical, because I do not know how many cases they are using this VAM inoculation or that's a mycorrhizal inoculation and also the biostimulants, so that they can easily acclimatize. Because that is one of the issue is acclimatization of the tissue culture plants to the environment. Because unlike other herbivorous plants, it is a very difficult issue.
Thank you so much, sir. We have noted your suggestion. Dr. Reddy is very senior expert, as I told. And some other suggestion you have posted, we will act upon that. Thank you so much, sir.
Yeah, Dr. Shukla, two important questions I want to take and address to particularly the industry expert, engineer Gautam and Balakrishna. One is what is the approximate budget to establish a commercial tissue culture lab? And second is a kind of complaint that nowadays most of the private companies are coming forward for sale of tissue culture plant to farmers on higher cost side. So first question, establishment, approximate establishment cost of the commercial tissue culture lab and the cost concern from the farmer's perspective. I would like to listen both of the experts from the industry.
Yes, sir. So actually, from 2006, we are into the industry. At that time, the banana was 10 rupees, and at that time, the labor was 80 rupees a day. Chemical was very cheaper. And even now, we are selling at 10 rupees the same banana, though the labor cost is 300 rupees. So everything has increased completely. And even the bamboos we are selling from 10 rupees to 30 rupees is the highest cost from our end. We are not charging more than 30. Basically, we believe this tissue culture, this agriculture, it is a service-based industry. So from our unit, I can guarantee you to all the farmers, we are not charging heavy. Maximum is 30 rupees. For banana is maximum is 10 rupees. And apart from that, the initial investment means it is depending upon the what scale they want. They want 1 lakh plants a year or 5 lakh plants a year or 1 million plants a year. So it depends upon that. The people can start with 1 laminar flow chambers as well. So the investment would be less than 5 lakhs to 40 lakhs is more than enough to scale up to 1 million plants a year.
Just to supplement what Mr. Palni said, cost of plant, it is the initial input. But the way the benefit comes, it compensates in a multiple way. So that is compensated by the benefit. Though cost of the initial planting material is higher in terms of tissue culture compared to the. Now setting up facility, just wanted to inform the audience that if you want to set up a good facility, the capacity should be at least 0.5 million, 5 lakhs plants. And for that, at least you need to invest at least 100 lakhs in Indian rupees. And for that, good subsidy scheme is available with the government of India, 50% of the total project cost. So that you can explore. Even NHB, National Horticulture Board, MIDH, Mission for Integrated Development of Horticulture, they provide 50% of the total cost subject to maximum 1 crore. That is capping. So if you opt for that, within 2 to 3 years, you will get a break even. So that is the option.
So, Dr. Balakrishna, what is the cost of the teak tissue culture plant ready to transplant?
Yes, sir. First, I will tell you, when I worked for Netherlands government, if we have land and building, we can establish one tissue culture laboratory with 40 to 50 lakh rupees. Maybe we'll produce 1 lakh plants from that laboratory. So with the land, if we have land, we can establish with 40 lakh rupees, first one. Second one is, here we are, selling at the rate of 300 rupees per plant, teak plant. In India, more than 10 companies, they are giving stumps in the name of tissue culture. They will give you at the rate of 10 rupees, 5 rupees, 2 rupees, and 150, 200 also. But they are stumps. We are giving genuine tissue culture teak plants and selling at the rate of 300 rupees per plant from other bioproduced plants only.
Okay, thank you. Thank you. I think it is very interesting and some more questions were there, but due to the paucity of time, I will refrain to take all those questions. And I hand over to Dr. Shukla for further proceeding because we are getting late already. Dr. Shukla, unmute, please.
Thank you, sir. We are receiving multiple queries as we told. So I request participant, if you wish to answer your query, please write to us. Definitely, as the organizer, collectively, we'll try to address your questions. So thank you so much for your response. Now we have come to the final segment of this webinar. And now I request Dr. Rishi Tyagi, as I told, he is the key person behind all this concept. So let's in the webinar series. So Dr. Tyagi, over to you for a brief closing remark.
Oh, okay. Thank you so much, Dr. Shukla. And we have, of course, we are conscious about the time and we extended too much today. So much interest has been generated. And we have the distinguished speakers and panelists who have shared their experience. I think that is quite motivating to the participants when they raise the questions from academic participants' side or from the commercial point. In fact, it has motivated me personally also because I come from a farmer family from India in the Western UP. And I am prompted to take up the plantation in teak, and maybe Dr. Balakrishnan, I'll be contacting you how you can help in that way. Like this, so many other participants and young participants must have been created the interest. And that is one of the purpose of this webinar, in fact, to create the awareness, what the Mr. Chairperson has mentioned in the morning, the 2-3 points, that awareness, and particularly in the regional languages, demonstration, and the cost effectiveness and hand-holding. So there are 3 key points I picked up from his speech. And purpose of these webinar, the ultimate is that we have to develop a program at the regional level where we can start the capacity building, including the awareness or the hand-holding to promote this tissue culture technology in various economically important crops. So that's the purpose of this whole series of the webinar. That's why you are troubled to give the poll questions to have the nerves to capture the nerve of the participant that what they think about this technology. So I will not take much time, but what I will say in my final words that this webinar and other webinars have been very satisfying to us as an organizer. And Dr. Shukla will equally also be very happy with this proceeding of the webinars, though it is extended with the time. What limit we fixed, but sometimes you cannot stop when you are having the interesting discussion. I apologize for all the speakers because they have been stayed back to respond to the queries very, very elaboratedly to the participants, whatever we could do. And I apologize to the participant that many of the questions, particularly the academic purpose, the media or the protocol, this we could not discuss it here, but nevertheless, the literature is available, and I would request participants to contact Dr. Shukla. He is very kind enough, And I want to tell you that the purpose is being fulfilled of these webinars, because many of the participants in previous webinar have contacted. They wanted to have the training on the tissue culture in different crops. And the next webinar we will be having on 26th August on the floriculture, or the ornamental crops, which is also the cash crop or the economically important crops in the region, in Asia Pacific region. So please stay with us till 26th August. And if you have not registered, you may register. You have the opportunity. And Dr. Shukla, I stop here by thanking to Mr. Charanjit Singh in absentia, Dr. Purnima Sharma from BCIL, from distinguished speakers and the panelists. I refrain to take the name because of paucity of time. And ultimately to Dr. Shukla, my very dear colleague, who has been taking a lot of pains to make these seminars very successful. And success of the seminar, I can rate it because there's so much interest is generated, and the speakers and panelists have the experience, shared the experience. I'm sure the many people from the different countries have been benefited. And let me assure you that the India is very progressive in case of adoption of the tissue culture technology in various crops. That's why you must have found out that the experts are the industry experts who are doing the commercial production and exporting the material have been requested, and they are kindly agreed to our request. So thank you, everyone. Thank you, BCIL. Thank you, ICRAF and C4 for organizing this webinar. Dr. Chandra is there. Thank you, Dr. Chandra, for coming forward. And the story doesn't end up here. As I mentioned, that we have to develop a program on capacity development, and maybe the industry people, Mr. Gautam or the Balakrishna, in relation to agroforestry, will be contacted again, and to seek their advice and the guidance and experience to be shared with the other people in the country. Thank you so much. I am fine. I am satisfied. Dr. Shukla, over to you if you have to say something else. Otherwise, that's fine.
I have to say just a thanks to one and all, and the best wishes. Please join us for the next event. That is the ornamental plant, 26th August. So thank you. Namaskar.
Thank you. Bye-bye. Oh, sorry. If we can have the capture a photograph, I forgot to tell. Just a minute. If you can put on your videos, and I have colleague Jack is there. If he is there, he can capture the one photograph for our record.
This is Jack. Let's take it twice. First one.
Please put on your video on.
Okay, let's do it.
Ardendu, Ramalinga Reddy.
Okay, let's do the first one. 3, 2. All right, wait a second. Let me save it. All right, let's do it again. 3, 2. Okay, thank you.
Thank you. Thank you once again, and bye-bye.
So bye-bye.
Okay, thank you. Bye, everyone.
Thank you, everyone.
Bye-bye.
Bye-bye.